Share the sound and screen. So as always, we'll start the lab with a little bit of a lecture, and that'll help me catch up on Chapter 2. And then we will discuss Lab 2. Do remember to read textbook Chapter 3 through the end of the section on the compound light microscope. You will need that to do Labs 3 and 4. Any questions about what we're doing? Alright, let's close that. And I should have it here. We're just talking about it. I don't think we talked about this slide. But we did talk about this slide. All right, let's move on to the next slide, acid-base balance. Organisms need to maintain a fairly constant balance in their cellular acids and bases to remain healthy. You may have heard that called homeostasis. It's not particularly an important term for microbiology, but all organisms need to maintain their proper pH. The pH is, I discussed, the amount of hydrogen ions in solution, and it's actually measured as the minus log of the H plus ion concentration. And that is what the number of pH means. As I stated, a pH of 7 means that you can have one hydrogen ion in 10 to the 7th water molecules. So that's what that 7 means when we say pH 7. I already discussed that. I haven't discussed this though, and that is most organisms grow best near a neutral pH. So most microbes grow best between pH 6 and pH 8. And you could even be 6.5 to 7.5. Now there are exceptions, and we'll talk about that later in the term. There are some organisms which grow under acid conditions, for example. And that actually would be the bacteria living in your stomach. Any question about any of that? Can somebody confirm that you're hearing me and everything's okay?
I can hear you.
Okay, great.
Yeah, we can hear you.
All right. All right, let's talk a little bit about organic compounds. We're looking at various organic compounds in this slide, and you can see they can be very simple. This one's even more simple. Or they can be complex, like this one here. The basic structure of an organic chemical compound is it's going to be held together by carbon, holding the rest of the molecule together. And that's especially true for the hydrocarbons, which are only carbon and hydrogen, as shown here. I can't read the name of that. That's pentane. Okay. Anyways, it's held together by the carbon. And as you can see, carbon binds to 4 other atoms, best shown here. And that is why carbon is a unique molecule for life, because it can bind to 4 other things. Carbon doesn't have to be written out in a stick form. You can also write it out in a ring form. And here, every point is a carbon. We don't have them numbered. We don't have them labeled carbon. Wherever there's a point, there's a carbon. And between this one and that one, there's 2 hydrogen bonds, why there's a double bond there. Between this one, the 9th, and I think the 8th, there's 1 hydrogen bond. And wherever you don't have a carbon, you have to put in the element. And let me see if I can see that. that slide. I'm not seeing it in another slide. Let me look here. No, I'm not showing it. Wherever there is not a carbon, you have to put in the other atom. And occasionally you'll see that. Actually, glucose would be an example, or fructose, where they have an oxygen there. Let me see if I can blow that up. No, that's a 12. I was wondering if that was another atom. So all of these have carbons. All right. If it's not a carbon on the point, like I said, you got to put in what the atom is. These are ring structures, but organic compounds can be in a linear structure like this, or they can be in a ring structure. So organic compounds contain carbon and hydrogen. They also may contain oxygen, and then all of the other elements for life. But the main two are always carbon and hydrogen. And even oxygen is at low number in most organic compounds. Carbon can bond to 4 different atoms. You can be in a chain or a ring structure. The carbon is the skeleton backbone holding the molecule together. And on the molecule there can be functional groups. This will be a compound that gives the molecule different properties. For example, methanol has the functional group OH on it, and that gives the molecule many different properties than without the OH, which would be methane right here. For example, this is a gas. It does not dissolve in water. If you put on the OH group, making it methanol, this is now a liquid and it dissolves in water. Any question about that? So the functional groups influence the chemical behavior of organic compounds, and actually of any compounds. The functional groups are responsible for most of the chemical and physical properties of a particular organic compound or molecule. I talked about that. Let's talk about the important functional groups in the biological molecules. OH we already talked about. That is seen in alcohols, but it's also found in carbohydrates, like glucose has an OH group, and it's also found in lipids. There are other functional groups that I'm not going to talk about, but you do need to know all of the functional groups in the red box. There's the amino group, which is NH2, and you find the amino groups on amino acids and in proteins. There's a carboxyl group, the COOH group, and you find this on all amino acids, on proteins, organic acids, and in lipids. And then there's the phosphate group, the PO4 minus 2, and you find phosphate in molecules like DNA and RNA, as well as in ATP. Now, why do you think I want you to know the functional groups that are in the red boxes, like carboxyl and amino? Anybody?
Because they're biological molecules.
Yeah, these are the functional groups that are important in the biological molecule. And we'll talk a little bit about them when we're talking about the biological molecules. For example, amino acids have an amino group, shown here, and they have a carboxyl group on each amino acid. So this being a structure of an amino acid, and the R group can vary for what it is. But all amino acids have an amino group and a carboxyl group. Any question about any of that? And so know the functional groups in the red boxes because they're the important groups of the biological molecules. When we're talking about the organic compounds, it's important to realize that the small organic molecules can combine into large macromolecules. This is especially true for the biological molecules. The macromolecules are called polymers, and they consist of many small repeating molecules called monomers. So poly means many, mer meaning a unit, and monomer meaning one unit. Any question? With the organic chemical compounds, we can combine two monomers to make a polymer by taking off an OH group from one monomer and a hydrogen group from another monomer to make water, and then link directly this R group that's part of the organic compound to this R group. And that is a condensation reaction or a dehydration synthesis reaction where water is pulled out of the two molecules, and then the two molecules are linked together directly to make the polymer, and then water is also made. Dehydration synthesis or condensation reaction. Both of those mean the same thing. The opposite is you can take the polymer, add water to it, break the bond here between the two R groups, break the water, and put part of the water on one R group, the hydrogen on this R group, and the OH of the water on this R group to make the two polymers. That we call hydrolysis, the breaking of a larger molecule by the addition of water. And hydrolysis is the opposite of dehydration synthesis. So dehydration synthesis is used by cells to make the biological molecules, the macromolecules, such as a complex carbohydrate, a lipid, and a protein. And then when the body wants to use that macromolecule, it can start degrading it by a hydrolysis reaction. And I know you're all familiar with hydrolysis from the movie Back to the Future, where Marty McFly told his mother, "Ma, you sure know how to hydrate a pizza." And how she did it was she put that hucky puck of a pizza in the hydrolysis machine, which added water, and then you got hydrolysis to make the pizza. Any questions? All right. I'm sure you've all seen that. All right, so let's look at dehydration synthesis in a little more detail. We have a polymer, and we can react a monomer to it. OH comes off the monomer, hydrogen off the polymer, and then we can link that monomer onto the polymer to make it a larger polymer, and then water is made. And then you can reverse that by bringing in the water and breaking that bond between this monomer and that polymer, and the water breaks apart, so the OH adds onto the monomer, and then the H adds onto the polymer. Carbohydrates include things like sugar, starch, and other complex carbohydrates. For our cells, they are an important energy source, but carbohydrates are also part of the structure of cells, like you can find carbohydrates on the cell membrane. And they're important in nucleic acids. Carbohydrates consist of the atoms carbon, hydrogen, and oxygen, but it's a specific ratio, with the general formula being for each carbon, there's 2 hydrogen and 1 oxygen. And then this will repeat for the molecule. Like for glucose, this general formula will repeat 6 times. So glucose has 6 carbons, 12 hydrogens, and 6 oxygens. And that's also true for starch, but starch would have, oh gosh, maybe hundreds of repetitions of CH2O. Okay? Any questions about any of that? The carbohydrates can be a simple sugar or strings of the simple sugars linked together. We can classify the carbohydrates into 3 major groups based on their size. The monosaccharides, which is a 1-unit carbohydrate. The disaccharides, which are a 2-unit carbohydrate. And then the polysaccharides, which are a mini-unit polysaccharide, mini-unit carbohydrate. An example of a polysaccharide would be the molecule of starch. Okay? Any question about any of that? For an example of monosaccharides, we have glucose shown here and fructose shown here. Well, let me blow this up a little bit. Let's put that over here. Wherever there's a point, we have a carbon. And wherever there's not a carbon, you have to put the atom. So this oxygen is put here because it's not a carbon, and that is a point. And the same right there. And this is the ring structure of glucose or fructose. You could also draw them out in a linear structure. And actually, glucose, most of the molecules are found in the linear structure when we're looking at glucose as a table sugar, a dry table sugar. Fructose, on the other hand, most of the molecules are in a ring structure as shown here. And that's especially true if the fructose is dissolved in water. That if there are some linear structure of fructose, it will react with itself and form the ring structure. Both fructose and glucose have the chemical formula C6H12O6. They're both monosaccharides. They're both sweet, so they're both simple sugars. But fructose is much sweeter than glucose. Any question about that? The monosaccharides, also called simple sugars, have between 3 and 7 carbon atoms, but most of the monosaccharides have 5 to 6 carbons. And they're sweet, and they're all soluble in water. Why they're soluble in water is the OH groups as well as the O group. And there's a lot of OH groups on fructose and a fair number on glucose as well. There's 3, 4 on glucose. So these molecules give the molecule the property that they dissolve in water. You can break down sucrose, which is the combination of glucose and fructose put together, by adding water and undergoing hydrolysis to break the sucrose, which is table sugar, the sugar we have on our tables. You can break that down into fructose and glucose by hydrolysis. If you don't know, fructose is the sugar found in fruit, and glucose is the sugar found in your blood. Glucose is the base sugar, and so you can find glucose in many places. And glucose is the first sugar made by most plants, like sugarcane, and then they convert the glucose into sucrose. At least, I should say, sugar beets and sugarcane convert glucose into sucrose. I'm not sure how they do it, but they probably combine it with fructose. So the sugar beets and the sugarcane probably make fructose, and you'd think that I would know that, but to tell you the truth, I don't know. And other plants do make glucose when they're fixing carbon dioxide, but usually they convert this glucose into starch. And that's why potatoes and corn and most other plants are starchy, because they convert the glucose into starch. But sugar beets and sugarcane convert the glucose into sucrose. Any question about any of that? All right, let's move on. Carbohydrates include the polysaccharides, which we have only looked at the monosaccharides here and a disaccharide. The polysaccharides have many monosaccharides. The polysaccharides can be straight or branched. Starch is usually straight, a straight chain, although it can occasionally branch. It doesn't have very many branches if it does branch. And some of the starch molecules are totally straight, and all they are is glucose, one after another, hooked together. So all of these monosaccharides are hooked together to form starch. Some other examples of polysaccharides include glycogen, which is similar to starch, but it's found in animal cells, especially animal livers, dextran, and then cellulose. When we're talking about grass, the main structure in grass is the molecule cellulose, and all it is a polysaccharide of glucose. But obviously we, being humans, cannot digest cellulose. So all of these are polymers of glucose, but they're covalently bonded differently. And we'll look at that in a minute. I'm not going to talk about chitin, so I'll just mention that these are some starchy foods, and they mostly contain starch, this molecule here. When we look at the polysaccharides, and you don't have to memorize this chart, I just want to show you. Let me see if I can blow this up. I just want to show you that starch can be a straight chain, or it can be a chain which does branch occasionally, but not very often. Glycogen, on the other hand, is a molecule that branches more often, but it is repeating glucose one after the other. And like I said, you can find this in animal livers, glycogen. Cellulose is a molecule of glucose that grass has and other plants. And it too is a molecule with glucose hooked together one after the other. But you notice the chemical link, we're not going to talk about chemistry in that, is a slightly different link than this one here. It's an alpha 1,4 glycidic linkage, and this is a beta 1,4 glycidic linkage, and we're just going to say it's a different link. And what it is, let me see if I can move this over, is that the blue are covalent bonds between the hydrogen and the monomers. So each orange or red object is a monomer, and they're hooked together one after another, and they're hydrogen bonded in a row. And then the yellow link, not hydrogen bonded, they're covalently bonded in the blue line. And then the next row is hydrogen bonded, where this sugar hydrogen bonds to that sugar. And as you can see in cellulose, there's a lot of hydrogen bonding. Each sugar will hydrogen bond with its neighboring sugar on this row and with its neighboring sugar on this row. And that makes cellulose very difficult to digest because of the covalent bond and the hydrogen bonds, and that there's so many of them. As I mentioned, cows can eat cellulose and digest it because of the microbes in their stomach, which break down cellulose. All right, we'll talk about peptidoglycan in a later lesson. Let me see if I can blow that up. And peptidoglycan does have sugar moieties, meaning a carbohydrate moieties, monosaccharides, which repeat one after the other, these orange things. But they have cross links, which might be shown in blue and green, which are peptides, linking the sugar moieties together. And peptidoglycan is a molecule we'll talk about when we're talking about the cell wall of bacteria. Okay? Any question about any of that? All right, oops, I just moved that. Let's move on. And I think I'll stop right here. Our next slide is on lipids. And before I get started on that, we're just going to stop. And I'll start here in our next lesson, in our next lecture lesson. Any question about any of that? All right. So for the lab, we're going to be talking about Lab 2 today, aseptic transfers. Oh my gosh, I have to discuss the infectious disease project as well. Let me talk about the infectious disease project now. This is a project you're going to be working on, and you're going to choose a topic and get it okayed by me to do research on an infectious disease. And it will be due Saturday, August 14th. What you want to do is pick an infectious disease and then email it to me and ask me if this infectious disease is one that you can do your research project on. And if nobody has chosen that infectious disease, I will tell you yes. Or if it is not an infectious disease, I will tell you no, you cannot do your project on that disease. Okay? Any question about that? So before you do the infectious disease project, before you choose it, make sure you get it approved by me. And it will be approved on a first come, first served basis. So if there's a topic you really want to do, you want to send me an email right away on that topic. Okay? But I'm going to require that everybody do an infectious disease on a different disease. And it has to be infectious, so make sure it is a disease caused by a microorganism. All right? Now, you know an infectious disease is one that can move from one patient to another, but an infectious disease is a disease caused by a microorganism that a patient can get from the microorganism. So Clostridium botulinum is not normally transmissible from one patient to another. It's usually transmissible by a patient eating contaminated food, but it is considered an infectious disease. Everyone clear on that? Most cancers are not caused by a microorganism, so most cancers are not an infectious disease. Okay? I'm just trying to explain some differences. Let me pull up the infectious disease project and I will briefly go through it. I should probably not show this. I'm not sure if this is... Yeah, this is the topics for last term. So let's close that down. Let me see if it's in there. Nope. Let me go online, because I'm not seeing it here, and I've got it on my thumb drive, which I guess that'll work. I'll use the thumb drive. Take off my headphone for a moment. And let's go here. Let's see if it's there. I'm still having trouble finding the file. If I don't get it shortly, I'm going to say, oh, here it is. Well, let's go with the infectious disease project. So you're going to choose a research project that's on an infectious disease. This will be a 100-point project. Make sure you get it approved by me, and then do research to learn about the disease. Write up a written report. There must be a cover, which is not a cover page. It must have the title with the name of the disease, your name, the date, the course name. I do not want a cover page because that takes up so much room, and it's true I won't be printing it out. If I were to print it out, it would be an extra page I have to print. I probably won't print it out, but I don't want the cover page. What I want is for you to put the cover on the same page as the body of your text. So on page 1, you'll have the cover information, and then you'll have a space, and then you'll begin the body of your research paper. In the text of the body of your research paper, you must describe all aspects of the disease and put it in headings for the different categories, and I'll show you the categories in a minute. And the body of the paper will be worth 60 points. There will be 15 additional points for the grammar, the spelling, the flow, and the readability. So if you write a good paper, you'll get the 15 points. If you write a sloppy paper that has lots of grammar and what am I saying, logic errors or or something that's not clear what you're talking about, well, then you'll get deductions on these 15 points. The text should be written in your own words as much as possible. Any information that you get from somebody else should be put in direct quotes, and then you say where you got that information. If you don't do that, that would be plagiarism. Any information that you report in your paper that is not common knowledge, then you must give an in-text citation for where you got that information. Like, for example, if I were to make the statement that in the United States we have lots of HIV patients, that would be considered common knowledge. But if I were to state something like, in 2020, in the state of Washington, there were 10,001 HIV patients, that you would have to put in a citation, because that is not common knowledge. And by the way, I have no idea if what I just said about HIV in Washington was true or not. I'm just trying to give you an example. All right? So anything that is not common knowledge, you must give an in-text citation. If you're repeating in the paragraph items which are not common knowledge, you do not have to give, after each sentence, you do not have to give an in-text citation. If it's one after the other, you can do at the end of the paragraph a citation for the entire paragraph. Any question about that? But on the next paragraph, you should also give the in-text citation for the next paragraph. So for each sentence you give that's not common knowledge, if there's one after another in a paragraph, you don't have to give an in-text citation after each sentence. You give one at the end of the paragraph. Everyone clear on that? Now, if there is more than one in-text citation, meaning you're referring to more than one source, it's up to you how you want to break that up. You could, in the paragraph, give each source separately for each sentence, or at the end of the paragraph, you could give 2 or more sources. That's up to you how you want to handle that. How you do an in-text citation, I'm going to show you, because students frequently get this wrong. Not all students, but it's enough students that I'm going to show you, because this error happens all the time. So we have our sentence. I'm just going to put a line there for the sentence, and then you do an in-text citation. You give the last person's name, so I'm just saying Smith, and then you give the year, 2020, okay? And if it's 2 people, you give both names, Smith and Jones, just their last names. Oops, didn't spell that right. And then you give the year, 2021. Oops. And you put it in parentheses. If there's more than 2 authors, you go Smith et al. And then the year. We'll say 2019 here. Okay? Oops, I don't have that in bracket. So that's how you do an in-text citation. And for every in-text citation that's in the body of your text, at the end of the paper, you then have your references, and that could also be called citations. And here you give the full citation. And this for the Smith would be Smith Let's go John Paul, JP. And then you state the article. What would be a name of an article? The effect of global warming on malaria. Effect of global warming on spreading malaria, or whatever. Something on malaria. Effect of global warming on malaria. And then you give the source, Journal of Infectious Disease. The volume number, let's say 221. And then the page number. You don't go in page there, you just put the number, but you're getting the idea. Now, do I have everything? Smith, the effect of global warming, the article number, and then the journal. If it's a page, a book, you give a little bit different full citation. I want to make sure I've got everything. I don't have everything. I've got to have the year in here, so that was 2020. And then the article name that you got it from, the journal it's from. I suppose that should be a J, capital J. Volume number and the page number. If it's an internet source, you do the same. Come on. Oh, I don't have content enabled. And then you give the web page. Oh, let's go HTTP. That's not quite right, but you get the idea. www. I don't know. What's a good one to go? Center for Disease Control.gov. And then we're going to go malaria. And then there's probably a bunch of other stuff in the HTML page. And then end it. Any question? So an in-text citation is something brief, and for each in-text citation, it should be in the full citation at the end of the paper. The in-text citation will be worth 10 points. I will just read through it and make sure you've got an in-text citation about every place you need it. Generally, unless students are putting it almost nowhere, I won't mark you down. I've never marked anyone down for having too many in-text citations. And then you must have it in the correct format. And then the full citation should have all of the information. And the full citation should have all of the in-text citation. And let me see how many points. I think the in-text citation is worth 10 points. Yep, 10 points. And then the full citation I don't see how many. Oh, there it is there. Is worth 15 points. For your citations, you need to have you can use any format. I think MLA was the one the plagiarism quiz used, but it might have been APA. Your sources should be good, scientifically appropriate, and reputable sources. So if you give a reference to something like the Sun or the National Enquirer, I'm going to mark it down because that's not a reputable source. They publish a bunch of stuff which isn't true. And for good sources, I can usually just look at it. Government sources like the USDA, the Center for Disease Control, scientific journals, Nature, Science, any scientific published journal that's reviewed, popular magazines, Time, Newsweek, Scientific American. Textbooks are actually an excellent source. If you look at your textbook, you'll see that it has many reviewers. And the reviewers go through the textbook to make sure that the textbook is factual. Newspapers from a credible source are accepted, like the New York Times. I'm not sure of the Vancouver paper, so I'm just going to say the Oregonian. Okay, a reputable newspaper is acceptable. Reputable scientific television shows, like from the History Channel, PBS. You should be suspicious of anything that's from a religious, politically organized group or a privately funded publication, because they will have a bias and they will publish the bias and ignore, I don't know, I don't want to say the truth, but that's what I'm saying. They will ignore the truth to put forward their bias. So you should be careful of that. You should not use personal websites because anybody can put up anything on the web. College and university pages are generally okay, but if you take a college or university page that puts up student work, you should not use it. Okay? And students can make an error and they don't know everything, and many times they think they know, but they don't know. You should not use Wikipedia because anyone can change the content of Wikipedia. You may use Wikipedia to get background information, but don't put it in your citation. What you can do is look in Wikipedia for their sources and then cite their sources. Okay? And I know that anybody can change Wikipedia at any time because I actually sat down with another instructor and then we changed something about Charles Darwin. We chose that one because we figured that that one they would be watching and would be more likely to block it than, say, I don't know, almost anything else. Like if we went and changed a page on cheese, nobody's really going to care, okay? But on Charles Darwin, they watch that one because there are a lot of crackpots who put in, I don't know, strange things about Charles Darwin. And so Wikipedia continuously watches that page. But we were able to change that page. So Wikipedia, anybody can change anything at any time, so you should not use it as your primary source. All right, I mentioned you have to have the citations, and it'll be worth 15 points. The final document must be submitted as either a docx, rich text formatted doc, or a PDF format. You might be able to put in a write, which Microsoft Word can do as well. All right, so you should have in the paper a heading talking about the causative agent and talking about the different parts of the causative agent. You should have a heading discussing the history of that disease. You should have a heading talking about the epidemiology of that disease. You should have a heading talking about the pathology of that disease. You should have a heading talking about the response and treatment of that disease. You should have a heading talking about socio and political and economic considerations of that disease. I have a rubric for how I'll grade the paper. The cover information is 1 point. The causative agent, the information about the causative agent is 9 points. The history is 10 points. Epidemiology is 10 points. Pathology is 10 points. Response and treatment, 10 points. Socio, political, and economic issues are 10 points. The quality of the writing, the grammar, the spelling, the punctuation, the flow, the readability will be worth 15 points. In-text citations are worth 10 points. The literature citation page will be worth 15 points, and that's what I was looking for. You must have at least 4 reputable sources to gain all of your 15 points, as well as have the citation in the correct format. All right? Generally, students are pretty good about the literature citation page if they have it. The only mistake they will make is they will abbreviate. Instead of spelling out 5 different authors, they will say Smith et al. That is not allowed. In a formal citation paper, you should not abbreviate unless you have 7 or more authors, and then you have to spell out the first 6 names, and then you can abbreviate. Okay? Then you can say et al. After you've stated 6 authors in a paper. All right? And generally, the best thing to do is not to abbreviate. The total points are worth 100 points. And when you do it, you should look at this for how I'll grade the paper. All right, any question about the infectious disease research project? All right, it looks like everybody's clear on it. And that paper will be due August 14th. It will take you some time to do it, so you should get started soon. Doesn't have to be right away. And then when you have a project, you should email me as soon as you come up with the project. And if nobody else has picked that project, I will okay that project. Any question about that? All right, if not, let's move on to Lab 2. Let me pull off my thumb drive. Don't ask me why I'm wearing the headphone if I don't have it connected. I'm assuming you guys are hearing me okay, because I didn't have the headphone on or
Yes.
connected.
Yeah, we can hear you.
Okay. Yeah, the computer also records, but it's supposedly the sound is better if it comes through the headphone. All right, let's go to Lab 2. Now remember, when you're doing the lab, you should put your answers on the lab worksheet. It's a smaller document than the lab module. So only submit the worksheet, not the lab module. If everybody were to submit the lab module, my Canvas website would fill up. And then you wouldn't be able to submit other documents. So Lab Module 2, learning about aseptic transfers. Upon successful completion, you should be able to understand the purpose of using aseptic transfer techniques, be able to determine if aseptic transfer was used from the data provided, and define the following terms: ubiquitous, aseptic transfer, inoculation, and contaminant. In our local environment, microorganisms are ubiquitous. They're in the air, the skin, they're almost everywhere. This is problematic when we're working in a microbiology lab because we generally want to work with a pure culture. Louis Pasteur came up with the concept of pure cultures where you only have one species of one organism, and then you study it. And the test results you get from one species will be correct. If you had two species that you were studying and you ran a test, you wouldn't know which of the species was positive for that test, and so we wouldn't know if that species was really, that we were interested in, was really positive for that test. And that's why we have to work with peer cultures. And Louis Pasteur developed a technique for making a peer culture, but his technique was not very easy to getting the peer culture, and we'll talk a little bit more about that later. And he also developed the technique of using aseptic transfer to minimize the risk of contaminating your culture, adding accidental bacteria and other microbes to the culture so it's no longer pure. That's what we mean by a contaminated culture. Anyways, we'll learn how to make aseptic transfers in this lab. During the inoculum, never lay the test tube caps down on the workbench. And then when you take off the lid, never lay it down on the workbench. What you should do is hold on to both the cap in one hand or the lid in one hand, and then as best you can, keep the plate covered so that the bacteria falls down from the air. It'll fall on the cover of the plate and not into the media. Now obviously you can't totally keep the plate covered all the time. You do have to get into it and do things. And then with the test tube, when you're not using it, obviously put the cap on, but when you're using it, you hold on to the cap in one hand, and then you tilt the tube so that if something falls down from the air, it's less likely to go into the tube because we have it tilted, in which case something falling down right here is not going to get into the tube, and only a small area, something falling down right here, can get into the tube and then contaminate it. So you hold the tubes at an angle. If it's a liquid broth, you can't hold it at a 90-degree angle. You have to hold it, I don't know, something like a 45-degree angle. If it's a solid, you actually could put it at a 90-degree angle, and then there'd be almost no risk of contamination. To make an aseptic transfer, you inoculate or transfer cells to a new media that's a sterile media, and you make sure that everything is sterilized before we inoculate it to prevent the media from being contaminated. And here we're looking at different things that we can use to sterilize the loop that we move the bacteria from this tube into that tube. And there is a loop here. It's called the loop because it's got a plastic or rubber handle, a metal portion of the handle, and then a wire portion, and then a loop at the end for holding onto many different cells. And what we do is we heat this wire portion by putting it in a flame or in the Bacto incinerator. We do not put the metal portion or the plastic or rubber portion in the heat, because that can't be heated, otherwise you can't hold on to it. You should know the terms, so go through the terms of this lab. You're not physically going to be performing any of these activities, but you will be watching videos on these activities and learning from the videos. We expect that you will know and understand the material in these videos. In procedure 1, we're transferring a sterile liquid using a glass serological pipette. So we're transferring water which is supposed to be sterile, from one container into a media, and then looking to see if that transfer was done aseptically. In procedure 2, we're going to transfer a sterile paper disc using forceps to pick up the disc and put it into the media. And then watch the video on demonstrating proper aseptic techniques. When you're doing procedure 1, read through it here. You're going to be measuring the water in a pipette and then putting in 1 ml of water into sterile media. And in theory, the water is sterile and the pipette is sterile. And when you get the water, sorry, this keeps moving on me. When you get the water on the pipette, you'll notice that the water in the pipette, in any glass container like a Biuret, the water will form a meniscus. And where do you read the watermark on the meniscus? Do you read it at the top of the meniscus, at the bottom of the meniscus, or in the middle of the meniscus? What you do is, for the meniscus, you read the meniscus at the bottom of the meniscus. Any question about that? And the meniscus forms because water is attracted to the glass of the pipette, and so it will be pulled up by the glass. So read about procedure 1. Like I said, you're putting water into a sterile broth liquid media. And then we have a picture of the results of that. In procedure 2, you're doing something similar, transferring a sterile paper disc into a tube containing liquid nutrient broth. And then in procedure 3, you're transferring Serratia marcescens to a broth culture, that's a liquid culture. And then in procedure 4, you're transferring Serratia marcescens to an agar slant. That is a solid culture. And we have videos for procedure 3 and 4 to watch, how to correctly transfer a bacterial stock culture from a bacterial stock culture to a sterile broth and a sterile agar slant. Now you should note that the video, I think it's using E. coli, which is a non-colored bacteria. Serratia marcescens is a pigmented bacteria. In a solid culture, Serratia marcescens looks red. In a broth culture, Serratia marcescens looks pink. Okay? And that will aid you in your visual identification of the growth. And that's the color of the bacteria. I should mention that when you're growing in a broth culture, it is problematic because it's difficult to determine if contamination is occurring in a broth, because in the broth, all the bacteria will grow together and will mix up. All the cells will grow up. You won't see one area of the broth being white from a contaminant, and then the other area being a different color from the bacteria that were supposed to be growing. They're all going to grow together, and if there's a minor contaminant, you will not know that there's a minor contaminant in a broth culture because it's liquid. If there's a major contaminant, you will probably notice that something's wrong. The culture may have the wrong color or the wrong growth characteristics. You should properly label all tubes, and this procedure tells you how to do it. You should label on a piece of tape, a test tube. Use a piece of tape to put your name, the date, the type of medium, The incubation temperature, I always tell my students, is optional. And then the inoculum, what you're growing in the tube. On a plate, you can write directly on the plate. You don't have to write on a piece of tape. And we'll talk about that in the next lab. All right, on procedure 4, it's similar, except you're going to be transferring Serratia marcescens to a slant culture. And here we're looking at a slant culture. You can see that a slant has the agar in a slant. And because of the agar being there, it's a solid culture. What you do is you inoculate the bacteria on the slant. Contamination is much easier to detect on a solid culture because, let's say you're inoculating a yellow bacteria. If all of the growth is yellow, you know that you have no contaminant. If, however, most of the growth is yellow, but there's one region, like right here, which is not yellow, let's say it's white or off-white, that would be a contaminant, okay? So you can determine whether you have minor contamination on a solid culture. In a liquid culture, it's impossible to determine if you have minor contamination. So interpret your results, and this table will help you. For procedures 1 and 2, it tells you the result. If the nutrient broth is clear, then there's no organisms growing in the broth. And for procedures 1 and 2, the aseptic transfer was successful. If the nutrient broth is turbid, that means hazy or cloudy, organisms are growing in the liquid broth, and the aseptic transfer was unsuccessful. For procedures 3, if the nutrient broth is clear, no organisms are growing. If the nutrient broth is cloudy, then organisms are growing in the broth, and probably aseptic transfer was successful. You can say it's successful if the broth is the correct color, okay? So this is the results and the conclusions for procedures 1 and 2 and procedures 3. And then for procedure 4, the results and the conclusions. So use the table to write up your conclusions. And then here are the results. You're not going to be doing this results. We've done it for you and taken pictures. This tube here is for procedure 1. And this tube here is for procedure 2. This tube here is for procedure 3. And this tube here is for procedure 4. Okay? And then answer the questions. There is a question 0, which is actually numbered 0 in the worksheet. So answer that question and answer the question 1 and answer those in the worksheet. You must list 3 sources of error, and that's why we have the 3 hyphens there. Answer question 2, which is for procedure 2. Answer question 3 for procedure 3. And then answer question 4 for procedure 4. And then write in your answer on the worksheet and submit your worksheet. Any question on any of that? All right, if not, go ahead and get started on the lab. I will be here for as long as the lab goes to help you and answer any questions. And I think that's 8:20, but let me check here. Yep, I'll be here until 8:20 to answer any questions. So get started on the lab.
Thank you.
You're welcome.